So, welcome everyone to the new seminar from the Spanish Society of Bioinformatics and Computational Biology. It is my pleasure to introduce today to Rafael Barrero. He is a graduate in biotechnology from the Universidad Pablo de Olavide in Sevilla, and he pursued a master in bioinformatics and computational biology at the Universidad Autónoma de Madrid. He's finalizing his PhD in molecular biosciences, focusing on new omics technologies towards a multi-omics approach. He's currently doing his PhD at the proteomics lab, cardiovascular proteomics lab here at CEMIC. He completed a research stay at EMBOL in Heidelberg and won one of the awards for the best poster at the first Congress from the CEBIP. That's why we invited him, and we are very happy that he could share his work with all of us, not only in the Congress, with a broader audience today. Actually, he will be presenting that work, Breeding Omics, user-friendly web platforms for multi-omics integration in metabolomics. Before you go ahead, Rafa, be aware that you can make questions either by Zoom, or you could also ask using the chat of the YouTube channel. Both ways are fine. And yeah, the floor is yours. Thank you so much, Rafa.
Thank you, Fatima, for the kind introduction. In this seminar, as Fatima said, I would like to talk about multi-omics, specifically multi-omics integration for metabolomics, the data analysis. I will first outline the main challenges in this field, and then I will introduce a web-based platform that we have developed in our group to address these limitations of metabolomics and multi-omics. As you probably know, multi-omics integration has undergone significant advancements in recent years due to technological developments in next-generation sequencing and mass spectrometry. The simultaneous probing of multiple omic layers within a single study has proven highly effective for the molecular characterization of multifactorial diseases, the discovery of novel disease biomarkers, and the advancement of precision medicine. However, despite the availability of various bioinformatic tools designed for multi-omics integration, important limitations persist when including in this integrative analysis data from other omics. And this is the case of metabolomics. First, metabolomics aims at measuring the metabolites present within a cell, tissue, or organism at a specific time and under particular conditions. There are two main strategies in metabolomics, which are targeted and untargeted metabolomics. Targeted metabolomics aims at identifying and quantifying a limited number of known metabolites, while untargeted metabolomics aims at acquiring data for as many molecular species as possible. There are significant differences between both methodologies. For instance, targeted approaches used in hypothesis-driven experiments and allows for absolute quantification of the metabolites, whereas untargeted metabolomics approach is employed in hypothesis generating experiments and relies on relative quantification. However, a key difference between these two approaches and that directly impacts in their integration with other omics is the identification reliability. In targeted strategy, metabolites are unambiguously identified, so they can be mapped in knowledge bases for example, for enrichment analysis, whereas untargeted metabolomics often relies on qualitative identifications with varying degrees of confidence. Actually, metabolite identification remains the primary bottleneck of this discipline. The question is, why is identification so challenging in untargeted metabolomics? The most commonly used platform for untargeted metabolomics is liquid chromatography coupled to mass spectrometry using the electrospray ionization as the ionization technique. In simple terms, analytes in the biological samples are first separated based on their physicochemical properties using the chromatography column, and then the monoisotopic mass of the analytes leaving the column at different retention times is measured using mass spectrometry. So the first step in metabolite identification involves searching the experimental monoisotopic mass the experimental monoisotopic mass search against reference databases. So this is the experimental mass that we have detected using the mass spectrometry, and with different tools, web tools, we can obtain multiple molecular formulas and candidate metabolites that may have generated this mass that we have detected with the technological platform. The point is that the researchers must manually review and prioritize these candidate metabolites before proceeding with the biological interpretation. So this is a complex and time-consuming process that greatly hinders the multi-omics integration of untargeted metabolomics data. It is true that several bioinformatic tools have been developed to perform multi-omics integration, like OmicsNet, or Omics Analyst, or PaintOmics, but these tools often fall short when analyzing untargeted metabolomics data. And these limitations arise from the inability to address identification challenges in untargeted metabolomics. But these tools also they also have limitations when analyzing targeted metabolomics data, because they rely on databases. They usually lack multivariate statistical techniques that may be very useful also in this context. So to address this issue, in our group, we have developed Turbomics, which is a web-based platform for multi-omics analysis of metabolomics, proteomics, and transcriptomics data. Actually, this platform is already available, and you can access using this link or this QR code. The point is that Turbomics accepts transcriptomics, proteomics, and metabolomics data collected from the same set of biological samples. And for metabolomics, the platform accepts or supports both targeted and untargeted approaches. Within Turbomics, we can automatically obtain the candidate metabolites using Seumas mediator, and the results, which are these candidate metabolites in the table that I showed before, can be simplified using turboputative. Turboputative is another web-based platform previously developed in our group to automatically prioritize the candidate metabolites generated in the database search. So the functionality of turboputative is detailed in this manuscript, but very briefly, it groups isomer and stereoisomeric annotations of the same chemical entity, and it also supplies a multi-criteria scoring algorithm that is based on signal correlation to rank the annotations. So the aim of this process is to make the matrix with candidate metabolites more suitable for the subsequent multi-omics integration. But of course, the user can also provide their own identification if they don't want to obtain them using Turbomics. After data preprocessing, the users can perform the exploratory data analysis and also apply multi-omics analysis using the multi-omics factor analysis, pathway integrative analysis, and enrichment analysis modules that are included within the web-based platform. In the next slides, I will show you these different sections of the platform, and I will also try to explain the underlying principles of multi-omics integration algorithms implemented, mainly MOFA and PathIntegrate. We have shown great results in metabolomics. This is the main page of Turbomics. Specifically, this is the new job section for data uploading, but the platform also has a find job section where the user can find previous jobs using an identifier generated by the platform. And this is the results section for the multi-omics integration analysis. Later, I will show you this section in detail. Within this new job section, the users have to upload upload the data, Turbomics utilizes a data frame system that allows orderly storage of the experimental data, facilitating data handling and management. Hence, a user have to upload a sample metadata table indicating the treatments, the conditions, or variables of interest. The user can also upload a metadata, omic metadata table. In this case, it is metabolomic metadata, but there's another slot for proteomics and transcriptomics metadata, where the user can indicate the variables or information of interest for the corresponding biomolecules. For example, the protein name, the lipid class. Here we can find the retention time, the ionization mode. This information can be really interesting and useful for the exploration and the multi-omics integration. And finally, the user have to upload the quantitative table containing a quantitative value of each biomolecule on each analyzed sample. Within Turbomics, these tables can be uploaded using a tab-separated values format, but this data can also be uploaded using multi-assay experiment class object. Actually, the user have to upload an RGS file containing a single multi-assay experiment class object, which defines a structure for storing and handling multi-omics experiment. It is a kind of generalization of the summarized experiment object of bioconductor, but for multi-omics experiments. Turbomics offers some functionalities and basic algorithms for preprocessing the uploaded quantitative data. The preprocessing is applied independently to each omic layer, and it involves three consecutive steps, which are normalization, scaling, and handling of the missing values. For now, we only have these normalization methods, though there is still work to do here, but in the future we will include some other normalization methods, for example the TMM, which is quite extended in transcriptomics. Another important issue is that different omics datasets often exhibit different value ranges due to the specific technological platform and also the normalization techniques applied during processing, and these differences can compromise the multi-omics integration, leading to distorted visualizations or hindering the interpretation of quantitative data. An example could be the representation using heat map. But we have to consider also that many multivariate statistical techniques employed in multi-omics integration rely on covariance maximizations. So this reliance on the covariance matrix for the maximization process can lead to an overrepresentation of datasets with larger value ranges, potentially overshadowing biologically significant patterns in datasets with smaller value ranges. So this is why we offer the possibility, and we also recommend the centering and scaling of the data. And finally, since many machine learning and multivariate statistical techniques cannot process datasets with missing values or managing missing data, this process or this step is crucial in the integration and analysis of multi-omics datasets. One common approach involves working with subsets of subjects or variables with no missing values, or alternatively, missing values can be addressed through imputation, which involves estimating and replacing these missing values with plausible approximations. So Turbomics combines both strategies to balance information retention and imputation. So users can interactively select a missing value fraction threshold and observe in real time the number of retained biomolecules. So after filtering, users can choose from several techniques to impute missing values of the remaining biomolecules. One major novelty, as I mentioned, of Turbomics is the functionalities for handling identifications and putative annotations, these candidate metabolites that I explained before. As I mentioned in the introduction, this is especially relevant in untargeted metabolomics. So there are two, within Turbomics, there are two possibilities. Users can upload their own identifications in the metabolomic metadata table, but users can also automatically obtain putative annotations using Turbomics, which automatically executes SEUMAS mediator and turboputative to provide this candidate metabolite. So in this case, of obtaining putative annotations using Turbomics, the metabolomics metadata table must contain the mass to charge ratio, as you can see here, the ionization mode, which is both of this information is used or are used by SEUMAS mediator to obtain the candidate metabolites. But this table should also contain the retention time, which is used by turboputative to prioritize the candidate metabolites. Some other parameters like the tolerance or the different adducts can also be specified within the platform. The result sections of Turbomics is organized into submodules. Some are dedicated to exploratory data analysis and others to multi-omics integration. Each module provides diverse visualizations and interactive tables, enabling fast and intuitive data exploration. The exploratory data analysis module offers researchers an initial overview and visualization of the data uploaded to the platform. Within Turbomics, the exploratory data analysis is conducted independently for each omics dataset. You can see here, for example, metabolomics and proteomics. So these processes performed in parallel for each omic, and to facilitate this process, the platform includes two dedicated modules. One is the data distribution that you can see on the left pane, and the other is the principal component analysis, the PCA, in the right pane. Data distribution enables users to examine the distribution of quantitative data using density and box plots. Users can explore these distributions by distinguishing sample groups specified in the metadata. So here the user can select the group used to aggregate the samples. And also the user can explore these quantitative distributions for a specific metabolite or lipids or protein classes. In the PCA section, Turbomics calculates the first 10 principal components and performs univariate linear regressions between the component scores and the metadata variables, the percentage of variation explained by each component and the... Yeah, sorry. Oh. Okay. I think there is a kind of background noise. So, as I was saying, the p-values from the regressions are displayed in a color-coded table, allowing users to quickly assess associations between quantitative data and metadata, as well as to identify the component linked to the conditions of interest. Additionally, users can select the specific principal component for visualizing the sample scores within a scatter plot, and users can also explore biomolecule loadings through an interactive
evidentment,
in addition to the exploratory data analysis section, Turbomics includes another 3 modules which are designed for multi-omics integration. The first module is multi-omics factor analysis, but later I will show you also the pathway integrative analysis and enrichment analysis. The multi-omics factor analysis module enables an supervised and integrative decomposition of the quantitative data into latent factors, providing an interactive environment for factor exploration and biological interpretation. This module is built upon the MOFA model, multi-omics factor analysis, which is based indeed in the group factor analysis model. So very quickly, MOFA models a correlation structure among biomolecules from different omics layers and allows the identification of major access of quantitative variations across data modalities. So as shown here, the quantitative data, as you can see here in the left, is decomposed into uncovered latent factors that are linear combinations of the original variables, which in our case are biomolecules, transcripts, proteins, or metabolites. So by identifying the factors associated to the biological condition of interest, we can explore which are the biomolecules associated to this condition and biomolecules that are likely correlated among them. Within Turbomics, users can explore a color-coded table summarizing the factors calculated by MOFA. This table includes the percentage of variance explained by each factor on each omic dataset, along with the p-values of the linear regression between the factor scores and the user-provided metadata. So this allows a rapid identification of the factors associated with biological conditions of interest, and also the contribution of each omics dataset. So users can visualize the factor scores in these scattered plots, and the distribution of the factor loadings across each omic dataset can also be explored. The user can select the top N, or top 50 or top 100 biomolecule with highest positive or negative loadings for representation in a heat map. So this provides an intuitive representation of the correlation structure explained by the factor and its association with the biological condition of interest. Notably, by clicking on this button, Turbomics also allows a user to inspect which are the biomolecules most strongly associated with the factor. So here we can explore which are the biomolecules represented within the heat maps. And the platform performs an overrepresentation analysis for each omics dataset using Go, K, and Reactome knowledge bases. So this analysis identifies enriched functional categories, thereby facilitating the biological interpretation of the results. Multi-omics integration algorithms often generate results that are difficult to interpret in a functional biological context. So the idea of this second section of pathway integrative analysis is to address this challenge by performing pathway-based multi-omics integration, which enhances the interpretability and improves the detection of biologically meaningful pathway signals with greater power and sensitivity. This module uses the PathIntegrate modeling framework, which is a Python library designed for the multi-omics integration at this pathway level. PathIntegrate condensed the molecular level measurements into pathway scores for each individual sample using dimensionality reduction technique and the information contained in knowledge bases. The dimensionality reduction technique that we use in Turbomics is the singular value decomposition. This pathway score matrix that is obtained is used as input to supervised models, which are multi-block PLS or PLS, the partial least square model, and this enables the identification of pathways associated with the outcome of interest. PathIntegrate can be executed in single view and multi-view mode. The multi-view approach calculates multiple pathway score, one pathway matrix score for each omics, whereas the single view concatenate the quantification, the quantitative data of each omic to calculate a single pathway matrix score. So within Turbomics, the user can select which kind of execution applies, single view or multi-view, the conditions that are going to be compared, and the name of the column containing the identifiers that are going to be used to map in the database. Upon execution, the platform displays a statistical summary table and a scatter plot, allowing users to explore the low-dimensional representation of the observations based on the calculated latent variables. And it can also be explored the most relevant pathways contributing to the model, which are potentially associated with the studied biological condition. So by selecting specific pathways, users can further explore which are the biomolecules associated to these pathways according to the knowledge base used. Turbomics also allow users to visualize a heat map displaying the quantitative data of biomolecules within the selected pathway. So this enables a direct visual assessment of how biomolecules within the pathway are associated with the biological condition under study. So this mitigates the abstraction and the interpretative challenges inherent to these kind of models based on dimensionality reduction, that they are often quite difficult, quite hard to interpret and to infer what is going on. And finally, the enrichment analysis module enables the identification of enriched functional categories for each omic dataset in parallel. So here again, you can see the metabolomics and proteomics. So this allows researchers to perform interpretable and flexible multi-omics functional analysis across different knowledge bases. So transcriptomics, proteomics, and targeted metabolomics enrichment analysis are performed using gene set enrichment analysis, using knowledge bases such as KEGG, Hallmark, GO, and Reactome. However, in the case of untargeted metabolomics, the enrichment analysis is performed using Mamichok library, which has proven highly effective for identifying significant metabolic pathways based on the mass charge ratio of the features. So biomolecules can be ranked using metrics such as log-fold change or t-statistic, but users can also incorporate PCA or MOFA factor loadings calculated within the platform. So this also enables a complementary insight into the multivariate models and enhancing the interpretability of biologically relevant factors. So to illustrate the ability of Turbomics in analyzing untargeted metabolomics data with a multi-omics approach, we have selected a previously published collaboration from our group. The aim of this study was to decipher the role of humoral immune response mediated by B cells in the development of atherosclerosis. So here it was found that the intravenous infusion of A12 autoantibody delayed the plaque formation and reduced the free cholesterol and LDL levels in this A12 treated group with respect to the other two control groups, PBS and B18. So the proteomics and metabolomics dataset of this study was reanalyzed using Turbomics. The proteomics dataset contained for more than 4,000 proteins, and the targeted metabolomics dataset contained almost 3,000 features. So the candidate metabolites were automatically obtained using Turbomics, and the multi-omics integration was performed using the multi-omics factor analysis section that I have just shown you. So the MOFA model could identify 8 factors explaining 68% of the variability in metabolomics and 76% in proteomics. And the factor treatment association analysis suggested that factor 4 and factor 6 were associated with the A12 treatment. We first explored proteins and metabolomics features associated with factor 4, and we found that these biomolecules exhibited a similar abundance pattern. They were highly correlated, and they showed a differential expression in the A12 treated group, specifically among the 50 metabolomic features with highest negative loadings that appear decreased in A12 group, were enriched in triglycerides, and they also showed some lysophosphatidyl including arachidonic acid that is involved in inflammatory process. And conversely, metabolomic features with positive loadings that appear increased in A12 group appear enriched in phosphatidylcholines that is described to have beneficial effects in the atherosclerosis. And consistently, the overrepresentation analysis of the top 50 proteins with highest positive loading revealed enrichment in lipid metabolism and fatty acid degradation. And with regard to factor number 6, among the 50 metabolomics feature with highest positive loadings, we found a highly presence of diglycerides and also triglycerides that again appear decreased in A12 group, and additionally among the protein with most negative loadings, which are increased in the A12 group, we found enriched categories related with protein translation and amino acid metabolism. So altogether, Turbomics provided results consistent with the original study, identifying significant alterations in arachidonic acid metabolism, reduction in triglyceride levels, and changes in the proteins linked to lipid and amino acid metabolism. And it is important to note that our analysis identified 37 decreased metabolomic features annotated by the platform as triglycerides and 27 diglycerides, which compared to the original study findings of 13 triglycerides and 3 diglycerides, it is a notable improvement. So this difference is due to the original study's approach of filtering for features showing statistically significant changes after multiple testing correction, which can lead to information loss. And this is an information loss that we do not suffer within the Turbomics environment. As I said. So to demonstrate Turbomics capability in analyzing targeted metabolomics data with a multi-omics approach, we have selected this COVID-19 study. In this study, the plasma multi-omics profiles in a large cohort of patients with varying infection severities are explored by targeted metabolomics and proteomics approaches. So the PCA of metabolomics and proteomics data reveals clear distinction of patient based on disease severity, and amino acids and lipids were strongly associated to the first principal component in metabolomics. So they show an interesting abundance pattern with respect to the disease severity. So there's a slight increase in the amino acids and slight decrease in the case of the lipids. And the multi-omics integration using multi-omics factor analysis module allowed the unsupervised identification of 2 complementary axes of variability highly associated to the disease severity. In the case of the first factor, it was found primarily associated with proteomics, as you can see. And it distinguishes healthy individuals from the diseased groups. So here, if we explore the proteomics or the proteins, we find NF-kappa beta signaling pathways such as CD40 ligand and ARAC1, and in the metabolomic side, we found some sphingolipids that were previously linked to COVID-19 severity. In contrast, factor 2 was associated with both omic layers, as you can see here, and differentiated moderate severe from healthy mild. So among the proteins that were increased with this disease severity, we found interleukin-6 and many pro-inflammatory cytokines, and among metabolites, we found ceramides and other sphingolipids. And we also applied pathway integrative analysis to perform a pathway-based multi-omics integration. So the comparisons were conducted in multi-view mode for the groups mild versus healthy, moderate versus mild, and severe versus moderate. So the most pronounced separations were observed in the mild versus healthy and moderate versus mild comparisons, suggesting greater alterations in proteomics and metabolomics profile during these stages. And all three comparisons revealed high prevalence of categories related to immune response, particularly interleukin signaling. For instance, a visual exploration of interleukin 4 and 13 signaling shows gradual increase in the levels of numerous cytokines from healthy to severe, with the most substantial increase occurring in the transition from mild to moderate. On the other hand, the most relevant metabolomic categories in the mild versus healthy were nucleotide metabolism, and in moderate versus mild amino acid and derivatives metabolism. So the original study analyzed the relationship between plasma proteins and metabolites using Spearman correlations. So the construction of protein metabolites, I mean, the analysis of protein metabolite interaction was performed using these regression models. So here we used a more comprehensive multivariate approach that enabled the detection of key associations between proteins and metabolite classes linked to COVID-19 severity. So we can reproduce the previous finding of the paper and also complement them with cutting-edge multivariate statistical techniques that project the data, the multi-omics data, into a lower dimensional space, capturing and highlighting the most significant sources of variations, and this in combination with the knowledge basis. So this is a summary slide of Turbomics. So, as you have seen, our goal was to develop a highly intuitive and interactive web-based platform that addresses some of the key challenges in metabolomics, particularly in untargeted metabolomics. We have incorporated modules and functionalities that we consider quite useful for a comprehensive analytical workflow. So this includes the exploratory data analysis, the statistical algorithms for multi-omics integration, and tools to facilitate the biological interpretation of the results. So as a conclusion, metabolomics is a rapidly emerging field and promising field in the molecular biology and personalized medicine. However, integrating metabolomics data within a multi-omics framework poses significant challenges, particularly in untargeted metabolomics, where the identification and characterization of detected features remain major bottlenecks. Turbomics is a web-based platform that offers the scientific community an interactive framework with the state-of-the-art algorithms for the analysis of metabolomics, proteomics, and transcriptomics using a multi-omics integrative approach. The platform includes functionalities to address key limitations in untargeted metabolomics, particularly challenges associated with putative annotations, enabling its analysis in a multi-omics context. Additionally, Turbomics supports targeted metabolomics analysis using framework where knowledge bases are combined with multivariate statistical models to identify functional pathways and abundance patterns of interest. And finally, by applying Turbomics to dataset from Lawrence and Japan, we demonstrate its ability to perform high-throughput multi-omics integration of metabolomics data, providing a more comprehensive view of molecular interactions. So this approach reveals correlation between molecular entities that might otherwise remain undetected, enhancing biological interpretation and hypothesis generation. So thanks to the cardiovascular proteomics laboratory, metabolomics platform, and lymphocyte biology here at CENIT, and also thanks to the quantitative biology and statistics group at EMBOL that also helped me to develop this platform. Of course, thank you all for your attention. If you have any question, I'll be happy to answer.
So thank you so much, Rafa. As we wait a couple of minutes to see if someone in YouTube have any questions, is there anyone here at Zoom that has any questions for Rafa? I will start, break
Okay.
the ice a little bit.
Yeah.
At the end, the integrative approach that you are using based on NMF is a semi-manual or semi-automatic one. It's not completely automatic. As many of the tools, for example, using deep generative models that are available now for single cell, DGM for Sarah Teichmann, some work from the group of Fabian Theis also. What would be your next? How would you develop further your tool to avoid this final manual step of trying to match the loadings, the factors with the sources of variability that you know?
Yeah, it's a great question, and I think this is the big challenge of bioinformatics applied to the development of tools for the scientific community. I mean, there is a big gap between the complexity of the multivariate statistical models or machine learning models and the scientific community in the sense that it is required knowledge in statistics and these fields of bioinformatics to apply correctly or to correctly apply these models. The way I try to solve or to address this gap is to apply the default parameters and also to make it in the same way flexible so that users can see different different biomolecules that may be proposed by the algorithms that are being used, and also to provide within the web page the links and manuals giving tips so that the user knows what is going on how to apply these models. But it is a difficult issue because when I'm developing the platform, I'm always like wondering whether the user is going to interpret correctly what is being shown. So it's not very easy, but the idea is trying to use default parameters proposed by the developers and also providing help pages and links to the source papers describing how to use these algorithms. Because otherwise, I don't know how to handle with it.
Any more questions? I was actually more thinking about the biological knowledge more than the statistical knowledge. That at the end, all these tools need a final step of knowledge about the experiment, knowledge about the
Yeah,
biology behind to interpret the results.
yeah,
So how can we go beyond, no, and do something completely automatic that doesn't require this final in the case, for example, of your problem, the final matching between the factor to a known source of variability that you have in your experiment. And
yeah,
I think this with deep generative models, that's a little bit what this is they are trying to do. That this final step is also automatic somehow.
yeah, yeah,
But of
yeah.
course, there you need because everything what you showed was bulk, wasn't it?
Yeah, it
What
was.
have you applied it to single cell data, for example?
No, I haven't. I haven't.
Hmm. Of course, you need more data for the deep generative models. But anyway, I love NMF, so it's
Yeah,
very nice work. Seems everyone is a little bit shy today. Um, these seminars are followed by many people just in YouTube because you can see them afterwards. Live is sometimes people are not able to make it Monday at 3, so many people watch them later.
okay, yeah,
So let's give a couple more minutes.
yeah.
Um,
In
also,
relation with what you said,
mm,
just that these tools are sometimes, or the way I think these kind of tools is for exploration. So it is what is usually said in the literature as hypothesis generating experiments. So maybe they are not useful for answering specific biological questions that may require very good knowledge in biology, but they can anyway be useful to give a first sight of what is changing, or which categories are changing within the studied biological system. So, I mean, well, maybe it is a good use of case for this kind of tools.
That sounds great. Okay, so again, Rafa, thank you so much. I think it's a great work, and that's why you got that prize in the Congress. So congratulations again, and thank you all. Probably some people will contact you by email when they watch online, and we will see each other in 1 month for the next seminar. Thank you,
Great.
everyone.
Thank you.
Bye.
Bye-bye.
Listo.